Hey, AP Bio Kids, this is Miss Hunt. I hope you're doing all right. I had quite a bit of you absent today. I had 18 kids in my two classes missing. Um, Make sure if you are quarantining, the school knows so they can mark you as quarantining. If you were just absent, a lot of unexcused absences, that's not cool. Make sure you guys are here and let the school know what your excuse is. All right, we're going to the lesson plans. I have us kind of sketched out until end of Christmas. So, all right, we are still in our unit 4 with the cell cycle. We're finishing this up. We had cell communication and cell cycle. So this week is cell cycle. We got a lot to get done. We're doing notes from Miss Woodfin, not the textbook. So if you want to look at the notes, you're welcome to go to chapter 12 and look at them. But nothing will be on Pearson this week. The end of the week, we have our test on the AP Classroom. The AP Classroom is doing something interesting. They, of course, have the videos that I've been putting up before, but they have questions now linked to just that video. So what I did is I made your quiz off of the questions with the video cue on them. So all the questions should be coming from the videos. So of course from notes in class and everything we're doing, but you should be watching those videos as a great way to study for your test. So make sure you're getting that done and preparing for your test on Friday. This is our last Friday off. Next Fridays, after Thanksgiving break, you'll be coming to school on Fridays. All right, today we went over some notes. We did mitosis sequencing, and we did not do the flip book. So I'm going to take that and see if I can scratch that out. Let's see, format, text, strike through. Okay, we ended up not doing the flip book today. Tonight, you need to watch the Bozeman video and if there's any other things we want to do. So we had these notes that are on the Google Classroom. We went through a lot of it. We didn't finish it. And then we did the sequencing sheet. Looks like this. And then we pulled this one too. Okay. We also did that one. So we kind of got a little bit ahead on that. Also, I gave you the Bozeman sheet. And that Bozeman sheet's worth extra credit, 2 points extra credit if you're watching the Bozeman video and doing that Bozeman sheet. Okay. All right. So let's go into our cell. Okay. That's what we're going to do in just a minute. Cell cycle division notes, this one right here. I'm pretty excited that we are teaching the cell cycle in honors biology too. So of course they take it on a much more simple level. AP Bio just assumes that you remember everything from honors biology, and that's not always the case. So sometimes we need to stop and remember where we are in the basics of everything. It is not bad to watch an Amoeba Sister on it, to YouTube something and go back and watch the basics of the cell cycle and about mitosis and meiosis. So if you need to go and do those things, by all means, do it, but make sure you understand. So eukaryotes, the heritable information is passed to the next generation through a process that includes the cell cycle and mitosis and meiosis plus fertilization. So we're going to begin by mitosis. All right, so here is how my prokaryotes reproduce. Remember, prokaryotes need to be able to communicate with each other so they know when to do this, when's a good time, and once they get that, enough of them, they can do that quorum sensing. So remember that chemicals communication between bacteria is how they communicate. All right, the cell here, first of all, do you remember what shape that is? If you remember bacilli? Remember bacilli, cocci, and spirillia? That's probably bacilli, kind of a rod shape. All right, the nucleus, there's no nucleus. So the DNA is going to unwind in a circle, and it unwinds and makes its copy at the same time. So it comes off like two separate circles in here. So it's going to copy its DNA. You can see that the cell is growing. It pinches apart. Cytokinesis, we have two. They are identical to the parent. Okay, there's no diversity here. One, that's good when we're talking about bacteria, because that means I have one strep test that I need to test for streptococcal bacteria, just one. There's one type of antibiotic unless the bacteria has become antibiotic resistant, but there's one method to treat it. So we like clones as far as medicine goes. As far as what's best for diversity and the survival of a species, no, sexual reproduction and diversity is way better. This could all die out. Unfortunately, it's not, but it could all die out if there was some kind of mutation or a weird temperature change, and they would all die. All right, for the cell cycle, for you to make a new copy of your eukaryotic cell, the cell has to accomplish a whole cell cycle, and that's what this big picture is. So this picture is showing interphase and then mitosis. So let's start with interphase. Interphase is the G1, the S, and the G2. So this is where the cell grows. It spends most of its time, like 60% of its time is spent in interphase. The S is when it synthesizes DNA. So it makes a new copy of the DNA. So you should remember the S is when the helicase unzips the DNA. You should remember DNA primase primes it, RNA polymerase builds it. Remember there's Okazaki fragments in the leading and lagging. Lagging had the Okazaki, and ligase glued the Okazaki fragments together. All right, G2 is when it grows again, and then it prepares for mitosis. So interphase, G1, S, and G2, the cell is doing its normal thing. What is it doing? Building proteins, protein synthesis. It is taking in nutrients. It's taking in amino acids. It's building proteins. It's doing cellular respiration. It's making ATP. It's letting out glucose. It's letting out ATP. It's letting out CO2. It's being a cell. Okay? The behavior of chromosomes during the cell cycle allows for heritability. So what it's doing is passing of these chromosomes on to the offspring. So that's what the chromosomes are. All right, before we get to a chromosome, we have to get to what a chromatin is. So our DNA gets wound up around a protein, and it gets wound up even tighter. And finally, it comes up to these chromatins. Okay, we'll see chromatin in just a minute. All right. There's some vocabulary that's a little tricky. We can do it. This is a chromosome, okay? Chromosomes are held together in the middle by a centromere. Here's the centromere is in the middle. All right, this is one chromosome. It is two chromatids, okay? We'll get to what that means in just a second. They are sister chromatids that get separated out into the new cells. All right, let's look at this one. This is one chromosome before mitosis. It's also one chromatid. When we talk about chromosomes, we're counting the centromeres. So this is one chromosome. This is one chromosome. And you're like, what? Why is that one chromosome? Because it is. Okay? Because we count the centromeres. Because everything wants to be more difficult than it has to be. That's science. Okay? All right, let's use, trying to find some good colors here. Yay. Okay. So you have 23 pairs of chromosomes. Which means you have 46 chromosomes. Okay. Let's take the 23 and let's think about chromosome pair number 1. Okay. So chromosome pair number 1. You get 1 from your dad and you get 1 from your mom. Okay. So 23 pairs of chromosomes make 46 chromosomes. So if this is chromosome number 1, I get 1 from my dad and 1 from my mom. Okay? So these are my chromosomes. And I would have 23 pairs. So this is 1, there'd be 2, 3. The last 23rd pair are your gender. It was also asked, where are these chromosomes? These chromosomes are in the nucleus of every cell. So every cell in my whole body, every nucleus has 46 chromosomes inside of it that are just me and my DNA. Okay? So in my gender ones, okay, if I am a girl, trying to do if I'm a boy here. Okay. So on my 23rd chromosome, I'm a girl. I inherited an X from my dad and an X from mom. I can't tell which is which. Okay, but I inherited an XX. Boys would inherit an XY. So boys inherited the X from their mom, and they inherited the Y from their son, or from their dad. I'm sorry, their dad. And boys will pass this Y on to their sons, or they pass an X on to their daughters. But if they have a boy, they know they gave that Y to make it a boy. Okay? So you either have one or the other for your gender. Okay? So these are in the nucleus of every cell. So we talked last week about blood and red blood cells. In every red blood cell, I have a nucleus with 23 pairs, 46 individual chromosomes. Before my cells, if my cells want to make new blood, maybe I was a donor and gave blood. If you donated blood, your blood needs to make more red blood cells from the old blood cells, then your red blood cells are going to replicate. And all 46 chromosomes need to go into the new blood. Okay? All right. This gets a little tricky. So this right here in the picture on your screen is one of these. So let's just say that's dad right there. Okay? Right here, where my finger is. I'm sorry, I'm holding it in 2 places. Right here. So this has to double, so I make sure I get my dad and my mom's information. So in interphase, in the S phase of interphase, everything's going to double. Okay, and looking for a black pen. Okay, so I have doubling of the chromatid. These are sister chromatids. That's still considered one chromosome because I count the centromere. This is considered one chromosome because I count the centromere. This is 2 chromatids, the sister chromatids. Okay? So here in the picture, we have 1 chromosome, 2 chromatids, and then they separate, so I get the 1 back. So what's happening in my cells over here, my pictures? If I was just reproducing this 1 chromosome, where I had 1 blue and 1 red, They double, and then I'm left here with 2 daughter cells, one blue and one red, identical to my original one right here. Okay. All right. In this chart, you can see prophase, metaphase. We're at 46 chromosomes. That's because at anaphase, the chromosomes pull apart, and we have 2 separate centromeres. So we think that they have, or you say that they've doubled the chromosomes to 92. At the end of telophase, it pinches apart. We go back to 46 individual chromosomes. The chromatids stayed the same, 92, 1 and 2. 46 doubled is 92. In the end, you end up with half that number of chromatids. All right, this is a good picture for mitosis versus meiosis. All right, mitosis. Here is my chromosome from my mom and my dad. They're homologous, which means they're chromosome like pair number 1, or they're my gender, pair number 23. So they have the same information on it, but yeah, they have the same information. This is called diploid, having a full set of chromosomes. Haploid is having half the number of chromosomes. So G1 starts, S phase starts, we double it. So here's the sister chromatids, 2 of my mom, 2 of my dad. Going on, G2, going into mitosis. Mitosis has PMAT, prophase, metaphase, anaphase, telophase, pinches apart, cytokinesis. All right, here we have 2 daughter cells identical to the parent. They are diploid, the full set of chromosomes. Okay? All right, this word right here is meiosis, meiosis. When I say meiosis with a southern twang, it sounds like mitosis. So I don't say it the right way. I say meiosis, and I emphasize the E in meiosis. I recommend you to write the word sex, S-E-X, through meiosis, because you'll remember that these are your sex cells, your eggs and sperm. If you don't want to write sex through here, then put E-G-G for egg and S-P-E-R-M for sperm. So you're talking about eggs and sperm, and that's it, okay? And I know when people talk about sex, they immediately think of plants. And yes, we will talk about how plants have sex. That's very interesting. Okay, I'm sure y'all want to know about the pollen and all of the parts of the flower. Okay, all right, so down here is meiosis. So we start off with a diploid, full set of chromosomes. They're homologous, that's the same pair of chromosomes. The sister chromatids doubled in the S phase. All right, this we called crossing over. This happens when a piece of the mom's DNA and dad's DNA switch. Why does it happen? It just does. There's no rhyme or reason. There's no point. Nobody knows why. The whole real reason is genetic diversity. It makes sure the offspring are not identical to your parents. It gives diversity, which is a big thing. But how, why, when, it just happens. All right, we continue on to the homologous chromosomes, 1 mom, 1 dad, and meiosis repeats. So this guy will also repeat. So half the number. So we end up with 4 cells over here with meiosis. 1, 2, 3, 4 instead of 2. Meiosis has haploid, half the number of chromosomes. So this egg would be fertilized with a sperm and continue and have a full set of chromosomes. Okay? In your eggs or sperm, you have 23 chromosomes. Just half the number of chromosomes. So that way, when you reproduce, your child will have half your chromosomes and half your partner's chromosomes. We go through meiosis again the week after Thanksgiving, when we do genetics. So we'll go through all of this again. All right, mitosis. The DNA replicates, we go through phases, we end up with 2 diploid, full set of chromosomes, identical. All right, prophase. In prophase, my nucleus starts to disappear. The centrioles form at the poles, and our spindle fibers start to come. Nucleus has disappeared. This is prometaphase. Really, there are phases of prophase, but prometaphase is the in-between. The nucleus is falling apart. The chromosomes are kind of wild looking. They're starting to find their sister chromatid and condense. Sometimes they look like a bowl full of noodles in here. Spindle fibers are here, and then the centrioles are at the poles. All right, metaphase. Everybody's lined up in the middle. Metaphase is the easiest phase to recognize. Everybody's lined up in the middle. It looks like stitches on a football. Okay, sister chromatids are lined up in the middle, and spindle fibers are attached to the centromeres in the middle. Anaphase. Anaphase away. The sister chromatids are pulled apart, and they're moving towards the poles. Telophase. We have pinching going on here, and the nucleuses start to form. Cytokinesis. Oh, we have separation. We have separation. All right, funny things. When that pinching starts to form in the cell, it's called a cleavage furrow. That's what it really looks like in an electron microscope. Here you see the cleavage furrow coming where it pinches apart and has two daughter cells. Plant cells don't pinch. Plant cells form a cell plate that becomes the cell wall. So you start to see this formation of the cellulose material forming, and then that turns into the cell plate, and then the daughter cell forms. All right, time spent in each cycle. Here's how long a cell spends. This is basically 1 day, the number of cells counted. So in interphase, they spent 16 hours, most of their lifespan in interphase. Prophase is another long period of 3 hours. Metaphase is very short. Anaphase, telophase, and then of course we have cytokinesis. So this lets you know if I looked at a survey of cells, I would expect about 60% of them to be in interphase. Most should be in interphase, and then you see the other phases here. Okay. All right, that's as far as we got to today. We will be finishing this up. We still have more notes to do. There's a lot more to do, but this is as far as we got today. All right, then we did this sheet. So I'm going to hold it up for you in just a second. You can pause the video here and do it, and then go back through it, okay? So I've got the answers up here. You want to zoom in on that. We labeled each of the phases, and then we put them in order, in number order. We went over it in class. I can scan it and put it on the classroom for you. Okay, hopefully you can see that. And then we decided to do this one tonight. And we could label the whole phases in here. So you can see there's PMAT right there. We have G1, S, and G2, and then PMAT and cytokinesis. There's some questions on the back for us to go over. Okay. And we did not do the flip book because we felt like we kind of got it after this. Okay. All right. I will come back on tomorrow with a new video.